Hello, I'm Maruk Said, your microbiology guide. Let's explore in-depth micro techniques and uncover groundbreaking insight together on this educational journey into the fascinating realms of microbes.
Environmental Biotechnology Explained. Table of Contents. Introduction to Environmental Biotechnology. Environmental Biotechnology and Sustainable Development. Objectives of Environmental Biotechnology. Applications of Environmental Biotechnology. 1. Bioremediation. 2. Monitoring through biosensors. 3. Sustainable industrial processes through the use of enzymes. 4. Biofuels. 5. Bioplastics. 6. Biopesticides. Introduction to Environmental Biotechnology. Environmental biotechnology is the application of living organisms for the protection, improvement, and restoration of environmental quality. In other words, it is the use of biotechnology to solve environmental problems. The International Society for Environmental Biotechnology defines environmental biotechnology as the development, use, and regulation of biological systems for remediation of contaminated environments, including land, air, and water. Environment-friendly processes such as green manufacturing technologies and sustainable development. The environment is continuously threatened by human activities carried out every day. With the increasing global population, there is a rising use of chemicals, energy, non-renewable resources, which leads to a growth in environmental problems. Although efforts to prevent waste accumulation and promote recycling are increasing, environmental damage is still rising due to overconsumption of resources, large amounts of waste generation, unsustainable use of land. A partial remedy to these environmental issues can be achieved through environmental biotechnology techniques. These techniques involve the use of living organisms in hazardous waste treatment, pollution control. If you have watched till here, don't forget to subscribe to Family of Science Lovers. Press the bell icon button so you are always updated. Environmental biotechnology and sustainable development. Environmental biotechnology can make a major and meaningful contribution to sustainable development. It can be applied in various ways to detect pollutants in the environment, prevent the release, emission of pollutants, remediate, clean up pollutants already present in the environment, different types of wastes, including solid wastes, liquid wastes, gaseous wastes, can be modified through environmental biotechnology by recycling them to produce new useful products, purifying them so that the final byproducts are less harmful to the environment. The replacement of chemical materials and industrial processes with biological technologies can help to reduce environmental damage. Objectives of Environmental Biotechnology. The main aim of environmental biotechnology is to prevent, control, arrest, and reverse environmental degradation through the proper use of biotechnology, often in combination with other technologies. One objective is to adopt production processes that make the best possible use of natural resources by recycling biomass, recovering energy, minimizing the generation of waste. Another objective is to encourage the use of biotechnological techniques, especially in areas such as bioremediation of contaminated land and water, waste treatment, soil conservation, reforestation, afforestation, land rehabilitation. A further objective is to apply biotechnological processes and their products to protect the integrity of the environment with the goal of ensuring long-term ecological security. Download our microbiology study app made for researchers like you. Applications of environmental biotechnology. Environmental biotechnology includes a broad range of applications such as 1. Bioremediation. 2. Monitoring through biosensors. 3. Sustainable industrial processes through the use of enzymes. 4. Biofuels. 5. Bioplastics. 6. Biopesticides. 1. Bioremediation. Bioremediation is the use of living organisms to remove or detoxify pollutants that contaminate soil, water, or air and pose a threat to human health. It is also known by other terms such as biotreatment, bioreclamation, and biorestoration. Bioremediation is not a new technique. Microorganisms have long been used to remove organic matter and toxic chemicals from domestic sewage and industrial waste discharge. It has great potential for cleaning water and soil contaminated with hazardous pollutants, domestic wastes, and radioactive wastes. Most bioremediation processes use naturally occurring microorganisms to detect, filter, and remove toxic waste before it enters the environment or to clean up existing pollution. Genetically modified microorganisms are also used to degrade materials that are difficult to break down naturally. Bioremediation can be carried out in situ at the contaminated site, or ex situ in specialized reactors after removal of contaminated material. Successful microbial bioremediation requires a suitable environment, and conditions such as nutrients, terminal electron acceptors, O2/NO2, temperature, and moisture may need to be adjusted to support microbial growth and activity. Plants are also used in some cases. For example, sunflowers can remove cesium and strontium from contaminated areas. Bioremediation is applied in environmental maintenance through 1. Wastewater and industrial effluent treatment. 2. Soil and land treatment. 3. Air and waste gas management. 1. Treatment of wastewater. Wastewater treatment from septic tanks, industrial discharge, and runoff from dairy and poultry farms is one of the most common applications of environmental biotechnology. One method involves adding microorganisms along with nutrients that allow them to survive and multiply even in harsh wastewater environments. These microbes break down hazardous wastes, making them harmless. Treated water usually has less odor compared to untreated wastewater. Microorganisms can also be used to control algal growth in drinking water reservoirs, irrigation canals, hydroelectric plants, and ponds. Introduced microbes compete with algae for nutrients and produce enzymes that break down algal cell walls. As dead debris floats to the surface, microbes digest it, leading to clearer water. Similar bioremediation methods are used to manage oil spills, whether from shipwrecks or fuel leaks, using comparable biological approaches. 2. Soil and land treatment. The heavy use of fertilizers, agricultural chemicals, and waste disposal practices has increased concerns about soil pollution. Various fungi are used in soil bioremediation, Rhodotorula species. Converts benzaldehyde to benzyl alcohol. Candida species degrades formaldehyde. And Aspergillus niger breaks down tannins from tannery effluents, improving soil for plant growth. Soil bioremediation is considered safe, reliable, cost-effective, and environmentally friendly for degrading pollutants. It can be performed in situ or by removing soil for ex situ treatment. In situ methods include adding nutrients, microorganisms, and ventilation at the contaminated site. Ex situ methods include excavating soil and treating it as compost or in slurry bioreactors. Bioremediation of soil is often less expensive than physical methods, and the byproducts are usually harmless. During treatment, soil microbes convert organic pollutants into carbon dioxide, water, and biomass under both aerobic and anaerobic conditions. Soil treatment can also be done using bioreactors, and the microbes used may be natural or genetically engineered. 3. Air and waste gases treatment. Air pollution mainly results from human activities, especially the burning of fossil fuels in industries and vehicles. Incomplete fuel combustion releases volatile organic compounds into the air. Landfills and waste disposal sites emit methane, and many household products also release volatile organic compounds. Initially, biological air treatment seemed impossible, but advancements in biological waste gas purification technologies made it feasible. Technologies such as biofilters and membrane bioreactors are now used to treat polluted air. Contaminated air is passed through bioreactors, where volatile compounds move from the gas phase to the liquid phase. A microbial community of bacteria, fungi, and protozoa grows in the liquid phase and removes pollutants. Bacteria commonly used for this purpose include Nocardia species and Xanthomonas species. 2. Environmental monitoring by biosensors. A wide variety of biological methods are used to detect pollution and for the continuous monitoring of pollutants in the environment. Biotechnology provides advanced techniques for diagnosing environmental problems and assessing normal environmental conditions, helping people become better informed about their surroundings. These methods are generally cheaper, faster, and portable compared to conventional laboratory analysis. Instead of collecting soil or water samples and sending them to a lab, scientists can measure contamination directly at the site and obtain immediate results. Biological detection methods using biosensors and immunoassays have been developed and are now commercially available. A biosensor may use a biological entity such as bacteria to monitor chemical levels, or it may use chemicals to detect biological entities such as pathogens. Immunoassays use labeled antibodies and enzymes to measure pollutant concentrations. When a pollutant is present, the antibody binds to it, and this interaction can be detected through color change, fluorescence, or radioactivity. Biosensors. A biosensor is an analytical device that converts a biological response into a measurable physical, chemical, or electrical signal. Most biosensors combine biological components with electronic systems and are often constructed on microchips. The biological element may be an enzyme, antibody, colony of bacteria, membrane, neural receptor, or even a whole organism. These biological components are immobilized on a substrate, and their properties change in response to environmental effects in ways that can be detected electronically or optically. Biosensors allow quantitative measurement of pollutants with very high precision and sensitivity. Current applications of biosensors include detecting toxin levels in ecosystems, identifying airborne pathogens such as anthrax, and monitoring blood glucose levels. Biosensors have been developed for detecting carbohydrates, organic acids, aromatic hydrocarbons, pesticides, and pathogenic bacteria. Saccharomyces cerevisiae, yeast, is used to detect cyanide in river water, while Selenastrum capricornatum, a green alga, is used for heavy metal detection. Many herbicides in river water can be detected using algal-based biosensors, where stress on the organisms is measured through changes in the optical properties of chlorophyll. 3. More sustainable industrial processes through the use of enzymes. The leather industry uses enzymes instead of harsh chemicals, traditionally employed in hide cleaning. In the textile industry, enzymes have replaced chemicals used for bleaching. The pulp and paper industry is reducing chlorine use through the application of enzymes. Washing powders contain grease and protein-digesting enzymes, which significantly reduce the amount of detergent required. Enzymes help lower the environmental impact of detergents because they save energy by enabling washing at lower temperatures, replace undesirable chemical compounds, do not interfere with sewage treatment processes, pose no risk to aquatic life, and are biodegradable. 4. Biofuels. About 86% of the world's energy comes from fossil fuels, and around 40% of this energy is from petroleum, most of which is used at the transportation sector. A biofuel is a fuel derived from plants or biological materials and is considered more environmentally friendly than conventional fuels because it releases less carbon dioxide into the atmosphere. Several types of biofuels are used as transportation fuels, including ethanol, biobutanol, mixed alcohols, biodiesel, and hydrogen. Governments worldwide promote biofuels because they are believed to reduce greenhouse gas emissions compared to fossil fuels. Ethanol is blended with gasoline in many fuels in North America. It is mainly produced from corn in the United States and sugarcane in Brazil. Biodiesel is a fuel produced from used cooking oil and other biological oils. Biogas is generated from gases released during the decomposition of organic matter in compost or landfills. 5. Bioplastics. Conventional plastic production from synthetic polymers consumes large amounts of non-renewable resources. These plastics create serious environmental problems because they are non-biodegradable. Bioplastics produced from plants help avoid the use of fossil fuels, require less energy and fewer resources, and contribute to the reduction of global greenhouse gas emissions. Microorganisms can be stimulated to produce enzymes that convert plant materials into the building blocks of biodegradable plastics. Polylactic acid is a biodegradable plastic that can be manufactured using existing equipment originally designed for petrochemical plastics. Polylactic acid has the second largest production volume among bioplastics. Polylactic acid is widely used in plastic films, bottles, and biodegradable medical devices such as screws, pins, and rods that degrade within 6 to 12 months. Manufacture of polylactic acid. Polylactic acid is produced from corn. Corn is sent to a corn milling plant and cooked for 30 to 40 hours at 122 degrees Fahrenheit. The softened corn kernels are then ground and screened to obtain cornstarch. Enzymes convert cornstarch into liquid dextrose, a sugar. The dextrose is piped to a lactic acid plant containing fermentation tanks. Microorganisms ferment the dextrose to produce lactic acid. Lactic acid is then transferred to the polylactic acid plant and heated to remove water. The product formed, known as a prepolymer, consists of short chains of about 10 lactic acid molecules. By increasing temperature and lowering pressure, lactide is formed, which is a ring-shaped compound created by joining two lactic acid units. Lactide is fed into a reactor where the rings are opened, making the ends highly reactive. These reactive ends join together to form long chains of lactic acid units, resulting in the polymer known as polylactic acid. Polylactic acid is sold in the form of beads, which are later processed into products such as cups, trays, films, and fibers. 6. Biopesticides. The excessive use of chemical herbicides, pesticides, fungicides, and fertilizers creates environmental hazards because many of them have low biodegradability. Biopesticides are derived from natural materials such as animals, plants, and microorganisms, bacteria, and they are considered less toxic than conventional chemical pesticides. According to the U.S. Environmental Protection Agency, USEPA, 2008, by the end of 2001, there were about 185 registered biopesticide active ingredients and 780 biopesticide products available. Biopesticides are classified into three main types: microbial pesticides, plant-incorporated protectants, and biochemical pesticides. Microbial pesticides contain a bacterium, fungus, or protozoan as the active ingredient that targets specific pests. Plant-incorporated protectants, PIPs, are genes and proteins introduced into plants through genetic engineering, enabling the genetically modified plant to protect itself from pests. An example of PIPs is plants that produce insecticidal proteins because genes from Bacillus thuringiensis, BT, have been inserted into their DNA. Biochemical pesticides are based on naturally occurring substances that control pests through non-toxic mechanisms, unlike synthetic chemical pesticides that directly kill pests. Biopesticides are effective in very small quantities and decompose quickly, which reduces their risk to humans and the environment. They are also generally non-toxic to non-target organisms. Thank you so much for watching. Don't forget to subscribe to my channel, press the bell icon button. Thank you!